These days, we hear less and less about HIV infections in our community. And that's because with current treatments, most HIV patients are living long, happy lives. So yes, we're winning big battles against HIV, but the war is far from over, and our drugs are not a cure. Take a look at this graph. When we start treatment here at the red line, we see the virus disappear from the blood. But even if we treat for years, once we stop the drugs here at the green line, HIV is back within 2 weeks. This says that the virus survives in cells of the body for decades even, protected. It's the reason we can't just pull patients off treatment to stop their drug side effects. And it's called the viral reservoir, and it's intensely debated. Now, the major reservoir are T cells, and they're very well studied because they're easy to get. You sample them for the blood and the red compartment at the top. But researchers in the field now agree that for reservoirs, tissue is the issue. And that's catchy, but what it means is that HIV in the blood penetrates deep into the body tissues, this sort of big proverbial black box here on the bottom. Down here, there are other cell types that can also be infected, but they've been largely ignored. Why? Well, they're a lot harder to access, right? We'll all agree to a blood sample, but if your doctor wants a chunk of your flesh, that's a different story. So how do we get tissue cells without a scalpel? Well, for my thesis, I work with HIV doctors in Malawi, Africa, where patients there undergo broncholavage, a technique to gently wash macrophages out of the lung. That's tissue cells, but no scalpel. But wait, HIV in the lung? Are you surprised? I was. But don't forget that all of the blood from the heart, all of it, goes straight into the lung for oxygen. And with that in mind, things can make a bit more sense. So I set out to learn whether HIV survives in macrophages in the lung during treatment. That's the definition of a reservoir. If we can show that macrophages are reservoirs, we can overnight change the way the field thinks about future treatments. It's such a privilege and really exciting to work on a project like this with a team connected to human patients. But what I found is that the tools to find HIV in T cells didn't work very well with macrophages. These cells just transmit the virus differently. So for my thesis, I generated some very powerful tools of my own called reporter cells that light up bright green once they become infected with HIV. And using these cells, we've mixed the reporter cells with patient macrophages, and something magic happened. Using the bright green signal, like a beacon, I can to light our way to the rare infected macrophages so we can study them more closely. And we've shown now that these macrophages do harbor HIV during treatment, just like a real reservoir. And ultimately, what we want to do is find ways to target specifically these tissue reservoirs and hopefully contribute to an HIV cure. Thank you very much.